All right, so we've talked about aerobic reactions. What is an aerobic reaction again? Requires oxygen. But you don't have oxygen in all cases. In this case, there are anaerobic reactions like fermentation. In cellular respiration, we took one molecule of ATP and one molecule of glucose and produced a ton of ATP. Roughly how much ATP was made from one molecule of glucose? 30, 34, 36 sometimes. So a lot. The big takeaway is a lot of ATP was made. But we absolutely needed oxygen. There are, though, cases where we might not have oxygen. There are cases where there's no mitochondria. Where might there not be any mitochondria? What organisms would have no mitochondria? Prokaryotes. So prokaryotes absolutely lack those internal compartments. So they cannot undergo aerobic cellular respiration. Yes? It's okay. There was a eukaryote found without mitochondria. And I guess now there's a lot of eukaryotes without. Turns out there are going to be some eukaryotes that lack mitochondria. Just because. What kind? So humans? Well, no. Humans have mitochondria. We absolutely do. But you remember endosymbiosis theory? Yeah. So with endosymbiosis theory, there were cells that had a nucleus. Some of those cells were able to take in a brown algae that became the mitochondrion. Does that make sense? That means that there's still other ones that had a nucleus, but no mitochondria. So they evolved separate than this other group. And this group just did really, really fantastically well, so much to the point that we just figured out that some cells don't have them a few years ago. Yes? There are lots and lots and lots of subgroups for eukaryotes. That's called taxonomy. It's something we'll study in 102 and something that drives me crazy because they keep changing it. Taxonomy. Taxidermy is different than taxonomy. Yeah. You're talking about taxidermy. Taxonomy is the science of classifying organisms. We can exist in a world that has no oxygen. There are times where you have no oxygen, or you might not. If you're underwater for a while, what happens to you? You drown. You die. Why? Because you lack oxygen. But there are obviously some organisms that can survive through those conditions. Has anybody ever done any canning by chance? All right, just you. That's okay. It's all right. It's only you. It's okay. Where I used to work, everybody did, because you have to survive the winter. With canning, you want to remove as much oxygen as possible. So the only things that can survive in there are the anaerobic organisms. With fermenting beer, you don't want oxygen there. You guys did a fermentation experiment already, right? What was the point? You had to get rid of that bubble. You had to get rid of the oxygen. So in order for these organisms to survive, the ones without oxygen or the ones that have no mitochondrion, they need to have a very fast and simple solution. Fermentation is an anaerobic process that's fast and simple, which is why I put up a picture of the Roadrunner, because he's both fast and simple. And I know it's weird, because I put this up and most people are like, I have no idea what that is. And then I feel, I know! And then I feel super old, and it sucks. Right. Okay, as long as you guys know. I appreciate it. There was a sign out in Arizona that said, coyotes live here. You don't have to. It was the police. They put out a sign that says coyotes live here. You don't have to put out a sign. You don't have to call us to tell us a coyote is present. Here are the steps you take to avoid coyotes. And it said, however, if the coyote is carrying a rocket or on skates or is dropping an anvil, then you should call us.
No.
And I appreciate that.
What you do is you do nothing. Eventually, it will hit something and it will bounce right back in it. You stand out of the way.
So fermentation converts glucose to energy without the need for other organelles. It's very, very simple and efficient. It is a two-step process. And the wonderful part for you guys is you already know half of it. You literally know the entire first step. It's just glycolysis again. The second step is a little bit more involved, but it's only one step. It's waste product formation. So step 1, glycolysis. I'm just going to draw it back up on the board. We take what molecule? Not trying to trick you. Glucose. We take glucose. And you break it in half to produce what? 2-pyruvate. I'll put a box around it. All right? What is that released? You've broken a bond, right? So a net total of 2 ADP go in and 2 ATP come out. 2 NAD plus go in, and 2 NADH come out. Plus. All right, so that's glycolysis. Questions on that? Now, that pyruvate from the next step can go on to become lots and lots and lots of different things. If it was exposed to a propionibacterium, you end up producing propionic acid and carbon dioxide. That's what gives Swiss cheese its bite. In case you didn't know, most of the cheeses and fermented things we have are made because of their interactions with microorganisms. Swiss cheese is well known because of what? The holes in it. So the holes in Swiss cheese are caused by bubbles of carbon dioxide. If you were to slice the cheese, you slice into a bubble, that's how you get a hole. If it was exposed to lactobacillus, we looked at lactobacillus in class. We took yogurt cultures and looked at those bacteria. If it's exposed to lactobacillus, it gives you the bite of lactic acid. Lactic acid is also what makes sour gummy worms sour. Lactic acid.
That doesn't sound healthy.
Well, most things that you eat, if you read the chemical formula or the chemical names of it, don't sound healthy. Dihydrogen monoxide. I mean, it sounds terrifying, but it's water. So it's like Saccharomyces. Saccharomyces are yeast. We use yeast for producing ethanol and bread. Thanks. You're the good one. Clostridium. Clostridium, if you're exposed to Clostridium, instead of making ethanol, you might accidentally make nail polish. Nail polish remover, rubbing alcohol. Or an Actobacter, or like E. coli, they go on to produce acetic acid, which we have in vinegar. This is the waste product.
Oh, okay, so they're doing this to make energy from stuff, then they expel this, and then we...
That's precisely what happens, and then we eat their waste products. That's exactly it. Do you guys know anybody that's ever tried to brew beer or something like that? All right, so this gets fun. They were so proud of themselves too when they first decanted it, weren't they? They're like, "You've got to try this," and they handed it to you, and you thought about it, and you're thinking, maybe I shouldn't. Few things happen to people who brew beer. One, if somebody hands you a beer that they've brewed, you need to trust that they did this with a very clean technique. That is, they were worried about the microbiology involved. A lot of times they aren't, but if they didn't wash their hands often enough, instead of yeast being used, it may be that they got an E. coli in there that turned all of their sugar in the beer into vinegar. It happens. If they did it wrong, you may end up with nail polish remover instead of alcohol. This is what happened to a lot of the moonshiners.
That's why you have to dispose of the first part.
Right.
Because it's
Exactly. So you know a lot about this. So with moonshining, if there was a small mistake, it could make people go blind or it could kill them. In fact, this just recently happened. There was a news article about this. Maybe one of you guys should find it for your journal. But there's a news article that in India, there were a group of people that were moonshining, and I think it was like 45 people were killed from it.
Yeah, that was recent.
Yeah.
That was like a week or 2 weeks ago.
So
Wait,
there,
how is that?
because the next article is going to be about this stuff.
Okay, cool. Thanks so much.
No problem. That's what I'm here for you. You know what? I'm fine with that. So you've got to be careful about when you're fermenting something to make sure you do it with a clean technique. You don't want to contaminate your samples. The first type of fermentation I want to talk about is a model that I think you all are familiar with, which is yeast. Yeast is going to take that glycolysis phase, break down pyruvate into a waste product. And the waste product in this case is going to be ethanol. So you're going to take pyruvate, you're going to cut off 2 carbon dioxides, and that makes ethanol. In the process, and this is the whole point of fermentation, you're going to convert NADH back to NAD+. Because that NAD+ is a rate-limiting step. It's a rate-limiting reactant.
Does it go under entropy?
It's not because of entropy. You're effectively creating a cycle here. Do you see what I'm saying?
That's what I was going to say. Does it lose any energy? Or can it just be continually?
You're going to recover that energy when you break that CO2. Do you see what I'm saying? So you're using that bond break to get that back.
How do you know that
C6H12O6 is glucose.
Do you know that formula?
Not really, no. Yes?
That's what yeast does?
This is what yeast does. Takes the pyruvate, cuts off carbon dioxide, and produces ethanol. So it's turning NADH back to NAD+. Because without NAD+, you don't get any ATP. Yes? Yep, yeast cuts carbon dioxide off, recovers NAD+, and produces ethanol. E. coli has another pathway that cuts something else off to create, not methanol. It creates, what was it I said ethanol created? It's like nail polish remover. Yeah, it's some bad stuff. Oh no, E. coli creates vinegar. It makes acetic acid. It cuts something else off to make acetic acid. Yes? I think so. If it's not, it's not. There is? Okay. So that's the whole process. Now this is kind of important and cool because we use these waste products. Carbon dioxide we use in breads. Carbon dioxide is what makes bread rise. We put yeast. Nope, it's carbon dioxide. Well, it's the carbon dioxide from the yeast in bread. If you're talking about the baking soda in like cookies, that's where that works. But in bread, you add yeast, and the yeast activates to produce carbon dioxide and waste, actually a little bit of ethanol. It goes away when you cook it. The ethanol is the waste. Have you guys ever baked bread before? Okay. Now when you're baking bread, see if we can tie this into some other things we've talked about. You're making bread. You take it and you beat it up for a while. You put yeast in there. Then what do you do with it after you've put all the ingredients together? You put it right in the oven? You let it sit, right? Yeah. At a warm temperature. Oh, you so got this. That's exactly it. So you put it at warm temperature. You put it at a warm temperature. You put it somewhere where it's nice and warm. And what that ends up doing is producing, as it gets warmer, you have higher kinetic energy, so you have more reactions happening. So there's more carbon dioxide made. There's more of that sugar that's broken down. What else do you do with it? Do you just like leave it in the bowl and put it somewhere to do its thing? You cover it. Why do you cover it? No, it's not the humidity. That's it. What about the environment around it you don't want it to react with? And this is important. Oxygen. You cover it in order to stop oxygen from interacting with it. Because the yeast are going to undergo aerobic cellular respiration until they don't have any oxygen. You cover it up, suddenly it starts doing this whole fermentation thing really quickly. I made a mistake once. I was like, I'm a biologist, I got this. I just need to completely seal it off so that it doesn't get any oxygen. So I took saran wrap and I wrapped the bread. Oh, I'm sorry, I wrapped the dough. Okay, then I set it someplace warm. So what happened? Why? You're right. It absolutely popped. It got bigger. Why did it get bigger? The fermentation produced what? Not oxygen. CO2, exactly. So it's building up to this big CO2 bubble. No, it just popped. But I do have some fun explosion stories. As far as CO2 buildup goes, you guys know some people who've tried to brew stuff, right? As they're brewing, the first mistake most people make is they don't have a release valve. That is something to release the carbon dioxide. So they have this big steel drum or this big metal drum that they filled with sugar and yeast and water. And they're reacting and producing a lot of carbon dioxide. So what happens to the pressure in it? It expands and expands and expands until what happens? It explodes. So there we go. Another fun story. I had two students who they told this story, so I thought it was very fun. You guys know what eggnog is, right? All right, so eggnog obviously has a bunch of sugar in it. Yeah? So they had this eggnog, and it was sitting for a while. And you guys have probably done this too. You've had bad milk, and you found bad milk, right? That's the worst, isn't it? Because you always find bad milk when you're eating cereal. It's you go to the refrigerator, you pull out the milk, you pour it into the cereal. It doesn't clump, because if it clumped, you wouldn't bite it. But it pours in, and you take a bite, and you're like, is it the cereal or is it me? And you have to think about it, and you're like, I'm going to take one more bite, because it might have been the cereal. And it's not, and you're like, great, now I'm eating spoiled milk. So you get rid of it all. What do you do with the milk? Right, that's what you should do. That is actually what you should do, but that's apparently not what I do. No, I don't want to deal with that right then. So what I do is I put it back in the refrigerator. No, I do that. And I know not to drink it. I put it in the back of the fridge.
But you forget it, where it is.
That's what happens, right? So has anybody ever forgotten an old milk in the fridge? Yeah? Okay, thanks. All right. For those of you who have, what happens to the container? It doesn't explode, luckily. If you look at, a gallon of milk, how it has those little bumps in it on the side? They expand out. That's one reason you have those bumps, so you know definitely when not to drink it. So I tend to get rid of it at the point where I want to get it out at trash day, because I don't want to open it. It's disgusting. I'll puke if I see the little chunks coming out in the sink. You know what I'm talking about? Oh yeah, it's terrible. I don't want to see it. I just want to throw it away. So I have never had it explode, but two of my students, they had eggnog over Christmas. They had it in their fridge. They said it was Easter. Easter rolled, yeah, right? Easter rolls around, they're like, oh, that eggnog's still in there. It was in a closed container. So they took it out of the fridge, and they're like, we have to take this to the dump. They didn't live somewhere they could just throw their trash on the curb and somebody takes it away. So they put it in their car with all the rest of their trash. Their nice, warm car. So what's happening to inside that container? Fermentation, heat, kinetic energy. It's moving faster. So they're driving along the highway, not the highway, they're driving the back roads because they're going to the dump, and they hit a pothole. And that pothole was just enough extra energy to snap the already strained bonds of the container. And they had a geyser of spoiled eggnog. Oh, yeah. At that point, there was no point trying to recover the car. It will smell like spoiled eggnog forever. Yeah. All because of CO2 buildup from ethanol.
Why you put milk back in the fridge if you knew it was bad? Yeah.
Okay, so I put milk back in the fridge because I know that I can't take it. I'm not going to pour it down the sink because I don't want to puke, and I want to get it into the trash. Well, trash day is coming in a few days. Close it quick. Look, you're just like my wife. You're just like.
You probably were just like, oh, I can put it back in the fridge and she can handle it.
That also could be a good idea.
That's what my brother
That never happens.
He's like, oh, Mom or Kate will handle it.
Sidebar, do you guys do the whole, I think this tastes bad. You should try it. Yeah, that's a horrible thing to do to people. All right, yes? No, it was in a plastic container. Oh, yeah, but it was on top, and you can't really close those bags all. You guys know a trash bag. You can't close them all the way. Yeah. So we produced ethanol. We got there through breaking down glucose to pyruvate, That pyruvate then breaks down into the waste product of ethanol. Questions? Seems a lot easier than that aerobic cellular respiration, right? Yes? Same thing I've been going over. Yeah, I've been saying the same thing like for the past 15 minutes. Please.
Like the energy from breaking the CO2 makes the NADH go back into NAD+.
Yeah, so the big thing is we have to recover this NAD+. So we're using that CO2, that NADH has a highly energetic electron, right? So between the product of the pyruvate and the energy from the NADH, you're able to produce the ethanol, which has a lot of energy in it. And that gives us that NAD+. We have to have NAD+ to power, and it's not even powering, in order for this reaction to happen.
NAD+ has less energy than NADH.
Correct.
How did it go less? Like,
Because you're transferring that energy to that molecule.
oh, wait.
Okay, so when you...
Where does the energy from breaking the CO2 go? Oh, so that just means a lot of energy.
That has a lot of energy.
The energy from the NADH and that one?
So think about ethanol, right? What have you heard as far as ethanol is used to do what? You don't know? Obviously, alcoholic beverages, but forget that. Have you heard of cars powered by ethanol?
Oh, yeah.
Yeah. That's where they get their power.
Okay, so a lot of energy.
Yeah. So fermentation produces 30 to 34 ATP. And actually, this is usually I put this on the exam. Fermentation produces 30 to 34 ATP. Cellular respiration produces 30 to 34 ATP. Fermentation produces 2. So where's the rest of the energy? It's in the process, but where is it here? That's exactly right. You store the energy in the bonds of the waste product. Partly, but more because it's got a ton of bonds in it. So you got lots and lots of bonds. They're not unstable, no.
I feel like that's like, I'm not saying it's like stupid, but it's like they just put so much energy into it just to waste it. Like, it's bad.
Yeah, well, you're burning this energy like crazy. And truthfully, think about it. If people want to work out and lose weight, they want to get rid of sugar. You want to push yourself to the point where you've used up your oxygen, and you've gone into a fermentative phase. Your muscle cells are termed facultative anaerobes. We'll get to them in a second. But effectively, you're going to burn sugar very, very quickly with no oxygen, because most of the energy is getting put into a waste product, not into powering your reactions.
Oh, yeah. Okay, that makes sense.
Another example is lactic acid fermentation. And okay, as far as this is, I guess, what people really want to know. What's the takeaway? The big takeaway for fermentation is you go from glucose to pyruvate, pyruvate to waste. Every single type of fermentation does the same thing.
The waste is different, and then the thing that gets cut off is different.
The waste is different, the thing that gets cut off is different, but the big picture is glucose, pyruvate, waste, recover NAD+. Yes, and then back. Yeah?
So would you say the reason why, like, only makes 2 ATP is the rest is you, like, how the other one comes to mind right now, where it makes 30 to 40?
I'm sorry, could you repeat that?
So would you say the reason why when fermentation only 2 is because the other one's like glycolysis?
Because all of the rest of that energy gets put into that waste product.
Yeah, because like,
Yes.
like the other one has oxygen to hold them together, kind of like a little bump.
Right.
More energy.
Yeah, that works. Yes, Brandon.
So the lactic acid fermentation, your muscles produce lactic acid, so is it caused by fermentation?
It absolutely is. So your muscles produce lactic acid. That is caused by fermentation. The point of any type of fermentation is break down glucose quick, recover energy. The point of any fermentation is break down glucose quickly and put that energy into the waste product.
And fermentation is the quickest way.
The fermentation is the quickest, simplest way. Like Brandon noted, some cells are able to do both aerobic cellular respiration and anaerobic fermentation. Your muscle cells are facultative anaerobes. They prefer to use oxygen, but when you don't have any oxygen, they switch over to fermentation. Your cells can't undergo fermentation for long because you're building up this waste product, lactic acid. But in a pinch, you can. That's why if you start drowning, you feel a burning sensation. That's why when you work out for a long time and you're out of breath, your muscles burn. You've got lactic acid buildup. The yeast that you worked with in lab are facultative anaerobes, which is why if you had a bubble at the top to begin with, an oxygen bubble, they used that oxygen first and didn't make as much carbon dioxide. They didn't undergo fermentation as much. So those are facultative anaerobes. There is also anaerobic respiration. That is respiration that occurs without oxygen. This is different than fermentation. Anaerobic respiration and fermentation are not the same thing. Anaerobic respiration is where you are undergoing chemiosmosis without oxygen. Fermentation uses substrate-level phosphorylation, an enzyme to connect a phosphate to an a phosphate to an ADP. Chemiosmosis uses ATP synthase. So if you look, we've got a bacteria here. I can get rid of this. We've got a bacteria here that's got invaginations. Like that. Sort of the membrane is folded inward. This is able to then create a hydrogen gradient in these little invaginations. So what happens is you have a pump that pushes hydrogen out of the cell. That pump is powered by an electron transport chain, just like with aerobic cellular respiration. So you're going to have NADH drop off an electron. That electron jumps and jumps. The difference is, instead of oxygen being the final electron acceptor, nitrate is the final electron acceptor, and it can convert it to nitrite.
Wait, so hydrogen pushed out, then nitrite comes in?
Nitrate is present, just like oxygen is present. Nitrate accepts the electron at the end, just like oxygen would, but instead of being turned into water, it's turned into nitrate. The big thing to note now, though, is it's a different electron acceptor at the end, but we've produced what here? What's that called? Lots of hydrogen. It's an electrochemical gradient. What way can these get back inside? With ATP synthase. So they go through, and that converts ADP into a TP. Yes?
So with the other cellular
Right.
the H plus ion was connected to the oxygen ion when it went through that process between after the cellular.
Right.
But with this one, you're saying that instead of the H connecting to the nitrate, it is going back into
The cell.
ATP.
Right. The other one, the hydrogen was still going through the ATP synthase.
Yeah,
So.
it was the leftovers from
Right.
that wasn't going in those.
Correct. Does it have anything to do with pH? You could say that the pH is constantly changing out here. The cell is pushing hydrogen out, but as fast as it's pushing hydrogen out, that hydrogen is going back in. So the pH isn't changing much. But it's a good call to connect them.
So, sorry, I know it sounds kind of weird.
Please.
This process is possible, like, what? It seems like it's this, maybe it probably yields a little bit less ATP.
It does yield less ATP.
Why is it that, like, how do I understand? How come
Because it comes down to a relationship that happened a long, long time ago. So there was an organism a long time ago that could use, so it looks sort of like this, right? And it's pushing hydrogen across and pulling it back in. Except it was using oxygen a long time ago.
What is nitrate? Like,
Nitrate's just, it's a chemical that is able to donate electrons.
so probably the reason why the oxygen ones survive more because they're just more oxygen.
At the time, yeah.
Okay.
So, but bear in mind, so you've got a lot of hydrogen being pushed out here. This is an organism from a long, long time ago. If this organism that was using oxygen as its final electron acceptor got eaten by a bigger organism, sort of like that, what have we formed if it wasn't digested? It's a double membrane bound organelle, specifically the mitochondrion.
Wait, so there are some mitochondria that don't use oxygen.
They're not mitochondria at that point. They're a different type of cell, a different type of prokaryote altogether. But if that cell would have been taken in by another cell,
These things don't exist anymore?
these things exist like, this is the oxygen one. This is the mitochondria.
Oh, no, I'm saying the one that uses the nitrate. That's what I'm
Those exist, but they're free living. They don't exist inside another cell
Oh,
that we know of.
Okay, sorry.
It's okay.
Curious about it.
So this process happens in organisms with no organelle. We're still moving hydrogen across a gradient. This is respiration. It's just respiration that happens without oxygen. You're using nitrite as that final electron acceptor. You're still pushing all of the hydrogen across the gradient. You're still accepting it through ATP synthase. You're still making a lot of ATP. It's just a different method. No oxygen's involved, so it is an anaerobic process. Remember, anaerobic respiration is very different than fermentation. Both lack oxygen, but that's where their similarities end. Any thoughts? Any concerns? All right, so the big takeaway. In today, we've taken a molecule, glucose, our model molecule, and we've broken it down. We've broken it down several different ways and several different times. We've taken this one molecule and we've cut it, and every time we cut it, we release energy. Remember, ultimately, this is all about oxidizing substrates, stealing electrons. We take the electron and we make it do some work. The work may be build ATP. The work may be recover NAD+. The work may be build a hydrogen ion gradient. But we're taking an electron from some other molecule, we're oxidizing that molecule, reducing some other molecule, and making that molecule do work for us. That's what we're all about. We need to take molecules, break them down, steal their electrons, and make the electrons do work.